modeling in the Office of Generic Drugs. His primary research interests include the application of quantitative pharmacology in product-specific guidance development and assessment. Thanks.
Thank you for the introduction. You know, obviously we need to communicate more about the ANDA. Well, I also like to thank attendees, not just in the room, but also joining through the WebEx from all around the world. And also thanks to the meeting organizer and Jeff Kelly's team, which allow us to know how many attendees in a very quantitative way. Yeah, thank you very much. And your attendance means a lot to us. And we are committed to advancing science and research and developing standards and guidances and then promoting generic medications. Now, presumably, you are a generic firm with an interest in developing some orally inhaled drug products. And you have taken notes throughout the presentation in the past 2 days. And you also read all the guidances, and then you take some important notes, for example, to understand your product and to follow some of the guidances we drafted. Now, so you did all the homework, and you read all through the piles of the guidance and then presentation slides. Now, you go over the list of your application submission, electronic files. You have the module 1, 2, 3. You know, now you go to module 5. So what module 5 is about is the clinical studies. And so oftentimes we do recommend the clinical PK studies because it's very sensitive to detect the formulation differences, if any, between the reference and your generic product. And so now I'm going to shift the gear just to talk about something which are not really existing in our product-specific guidances for the orally inhaled drug products. And the topic is batch-to-batch PK variability. So here is an outline of my talk. First, I'm going to introduce the batch-to-batch PK difference of the orally inhaled drug products. Second, I'm going to introduce some of the quantitative analysis of the potential factors contributing to the batch-to-batch PK difference, and then follow up with some recommendations on the test batch for PK studies. So as we all know, throughout the discussion in the past 2 days, complex product is defined in the GDUFA 2 commitment letter, and then orally inhaled drug products is part of the complex product. And take the fluticasone propionate, salmeterol-xenophosphate, orally inhaled powder as an example. It has a complex drug device component. It also has a specific absorption path to the delivery route, which makes the drug kind of locally acting in the lung. And so Adavir discus is a reference listed drug. And it's an inhaler consisting a combination of fluticasone propionate and salmeterol as a powder formulation for oral inhalation. And it is indicated for maintenance treatment of asthma in patients aged 4 years and older, as well as treatment of airflow obstruction and reducing exacerbation in patients with COPD. As I showed in the figure, the Adavir discus is a device which is a flat and a round, and it delivers the drug powder through the mouthpiece to the patient. And before each deliver application, the lever should be slided over to the other extent until one click sounds. And also, there is a counter which shows how many doses are left. And talk about the batch-to-batch PK difference. There is a paper published in 2016 and another paper in 2017 in the Clinical Pharmacology and Therapeutics mentioning about some of the PK studies using different batches in one study, and then to check whether the PK equivalence can be claimed between different batches of the reference listed drug. As I showed in this figure, this study was designed as a full treatment for sequence for period crossover study, and the full treatment group include one test product and then 3 different batches of the reference listed drug. And based on the publication, the study analyzed the batches of the reference product using the current BE assessment method. And as showing the figure on the left, it shows the Cmax of 2 APIs. And then in the middle shows the AUC from 0 to time t. And then on the right is a AUC for infinity. And the show. On the x-axis are the reference, the one batch over the other batch, means which are R1 over R2, R3 over R2, and R1 over R3. And the y-axis means the geometrical mean ratio with a confidence interval indicated by the error bar. And obviously, it is quite convincing that some of the batches does not pass the current B limit, which is 80% to 125%. For example, for fluticasone, Cmax, for the first 2 pairs, it's way over that limit. So this study basically suggests the PK difference is existing in some of the batches being tested in this study. So we are concerned about the paper result because this is a really critical issue when studies are designed to have a reference product batch as a reference standard to conduct a BE study for generic products. So we take a step further to look at the possible factors contributing to the batch-to-batch differences. And as you can imagine, there are certain differences in the API in the products among different batches. The storage condition as well as stability may also different. And then inactive ingredients from some source and may affect the quality. And then the drug performance, especially the single actuation content, as well as aerodynamic particle size distributions, may affect the PK as a whole. So we, and also think about the device application. It also involves some human factors as well as the variabilities among different applications. So based on the limited data we have in the agency, we did some preliminary analysis just to see whether some of the factors really contributed to the batch-to-batch PK differences. As I showed in this figure, we took the differences of the expiry date between each batches tested in the studies, and then we also plot the geometrical mean ratio against that expiry date difference, and then we can look for any of the variabilities that can be explained by the expiry date. So in the figure showing on the left panel, the x-axis means the difference of the expiry date between 2 batches. And there seems to be some variability of the GMR can be explained by the difference in the expiry date. However, we need more data and we need more information to draw any conclusion on that regard. In the next slides, we also expand our analysis to include some demographic information in those clinical studies. Those demographic informations include the age, body mass index, and weight. And we look at the potential correlations among the demographic informations and the geometrical mean ratio of 2 batches. And the study does not show any significance in terms of the variability explained by the age or BMI or weight overall. However, in some of the studies, we do see demographic information such as age may explain some of the batch-to-batch PK variabilities. So overall, we have very limited data in this area, which prohibits us to conclude any contributing factors at this moment. However, we do have a consistent recommendation on the B approaches when you consider the BE-PK studies. For example, in the current product-specific guidance, we recommend the firm conduct in vitro characterization using at least 3 batches, and then no fewer than 10 units from each test or reference listed drug batch. And then we also further recommend in vitro characterization in terms of the SAC and APSD. And when you have any alternative approaches before you conduct a study, welcome to contact OGD for guidance, either, for example, through a controlled correspondence. And finally, I like to conclude my talk by acknowledging people in the Office of the Research and the Standards. Thank you.